Jonathan, good to see you, and thank you for joining me. You know, I want to get a little history from you. So, what was your first introduction to echocardiography?
Yeah, well, it's great to be with you, Randy, and this is a special opportunity. So my first introduction, I was a resident. All residents are, when I trained in the, well, I shouldn't say when I trained, but I trained in the early 90s. We were exposed to echo, but I had no idea the breadth of the technology. And I think what really turned me on to echo and really opened the doors was reading a couple of papers it was from my future mentor, Sanjeev Kaul, about the study of the microcirculation using ultrasound contrast agents. And for a long time, I knew I was interested in physiology and in vascular biology, and really that was eye-opening in terms of what could be, what the possibilities were.
So you've been involved in, you just mentioned, a lot of technical developments in the field of echo. What are the things that stand out to you? Well,
some of the things that I've helped lead, and by I mean we.
Yeah, right.
Really, one of the things that I think that I've been able to contribute the most is getting young people excited about science in imaging. But some of the things that we've been kind of responsible for was starting the whole field of molecular imaging with ultrasound, essentially taking ultrasound contrast agents and saying, yeah, we can phenotype the microcirculation looking at flow and volume, but the ability to essentially put molecules onto the surface of bubbles to make them smart, to make them stick to disease, to be able to understand pathobiology. Where we're at right now with that is we're still using that technology less as a diagnostic and more for the process of drug discovery and drug development, to be able to do phenotypic imaging to usher in new drugs. The other thing that I've been heavily involved in is ultrasound therapy. Right. So gene therapy, drug therapy. Just got an NIH grant just 2 days ago on being able to deliver AAVs, adeno-associated virus, with ultrasound cavitation and energy. And right now we're in our very first human clinical trial for augmentation of tissue blood flow in peripheral arterial disease.
So really this is therapeutic usages of ultrasound.
That's exactly right. Taking advantage of the acoustic energy that comes out of oscillating microbubbles, either for increasing blood flow through shear-mediated pathways, or for essentially having beneficial bioeffects that help tissues of your body take up genes or drugs.
That's something that I'd always envisioned that we'd be able to do, and certainly sounds like you're reaching it. Now, you've had an incredible career, and I say that truly. You were the Young Investigator Award for the ASE. I think that was in like 1997. You also got the Richard Popp Award, which is phenomenal. But tell me something about how you got involved with the ASE.
Yeah, I got involved with the ASE. The first time I went to the ASE, it was in Toronto. I believe it was in 95, 96, something around there. And I knew I wanted to be involved in imaging and probably echo and do science. I had no idea the breadth. The other thing is I tailed people around. I had very good mentors, and I tailed them around. And it wasn't just the clinical excellence and the scientific excellence. It was the camaraderie. It was actually seeing how people, hugged each other, how they greeted each other, how it really was a family. And as soon as I saw that, immediately I identified myself as that's going to be a home for me.
I want to go back to the milestones of your career. Tell me, what do you've had a tremendous career, and I mean that sincerely. What are your highlights as you look back on your career to date?
Yeah, so several of them. Number one, the mentoring, the young investigators. I think I've had something along the lines of 8 or 9 young investigator finalists and many young investigator award winners within the society. And there is nothing more rewarding than watching the young people get engaged, the early career people really take advantage of everything that the society has. Other things that being president of the society was an incredible honor, and being able to work alongside, so many absolutely amazing people. The ASE is it has a way of essentially tapping into people's superpowers, their talents, whether it's the staff or the physician or sonographer volunteers. And so that has been absolutely incredible as well. I'm really proud of how much I've been able to bring science into the society as well. During my presidency, we had the E21 Awards, which funded physician engineering partnerships, and to this day continue to pioneer new avenues for research funding for early career people. And so those are some of my highlights. And then, of course, the research itself has taken me into just some incredible places in preclinical research, clinical research, animals, even astronauts.
Yeah, I mean, your career has been really dramatic, and I think all of those areas you've made have been tremendous as a physician leader, but also as mentoring people and getting people to grow. I did have a chance you were the 29th president of the society, which is fabulous, and I did have a chance to read your goodbye message, which was entitled "How Happy I Am." But the things that you talked about in your last message were really very important. Expanding those that were echocardiographers. I mean, you made big inroads into point of care ultrasound, veterinarian ultrasound, critical care. Tell me a little bit about how that came about.
Yeah, it essentially was a major focus of the presidency at that time, which is to address new users. A lot of people, especially, for example, in point of care, to some degree in the veterinary sciences, kind of saw the development of new users as a threat. We essentially, and by we I meant the executive committee at that time, because they worked together with me, saw it as more of a challenge and an opportunity, and an ability to essentially say the ASE is such a good institution for education and for engagement. Why not try to increase our impact by reaching out to these people? And so, for example, in the veterinary world, I am incredibly proud of the inroads that we made during my presidency to bring the veterinary imagers in, and they do incredible work. I think most human echocardiographers and cardiologists don't realize the sophistication, the dedication, and the skill of people who are doing small or large animal imaging and guiding of procedures.
Right, and you're exactly right about that. You've done critical care was another area that you got actively involved in and brought along. I mean, I think that's tremendous. You also did a lot of things in really marrying innovation and science together. Tell me a little bit about that.
Yeah, my entire career has been built on collaborative science. So when I started molecular imaging, it required me to go train under a vascular biologist. I didn't have any basic science vascular biology background. I needed to go learn vascular biology. I needed to learn basic physiology. And so there were the people there, when I was training and early faculty at University of Virginia that were world's experts in these areas. And so it really allowed me to kind of understand that. Then on the other side of things, the imaging technology, to be able to work side by side with engineers from a lot of the companies, it was a very special time. It's something that doesn't happen nearly enough right now. So much of our innovation in our society is somebody creates a new drug or a new button on an ultrasound machine and they say, here it is, go figure out how to use it. Back then, much more side-by-side engagement with people who understood clinical medicine, physiology and biology, and then engineering with the company engineers that would come and sit with you. To this day, I remember sitting there and for the very first time turning on something called harmonic imaging. And we had developed it for bubbles, and I remember everybody standing around looking at it and saying, wait a second, we can actually see the endocardial borders better with harmonic. And that was kind of the birth of harmonic imaging. And so that's something that I tried to do during to bring back some of the collaborative science between, especially engineers with industry and clinician scientists.
And you're so right that in the early days we did a lot of that because there was not the concerns about conflict of interest that it is now.
Right.
And you learn so much when you're wed to scientists who can. Both of you together can advance the field, and that's clearly what you've done.
Yeah, and this, I mean, you did a lot during your days, during the early days of ECHO. not to date you, Randy, but doing the same thing, which is side by side with the engineers of these companies to kind of figure out what 2D and Doppler were doing.
And color and all that.
Yep.
Jonathan, you've been an innovator, and that was another part of the thing that you did in your page. But let me get you to put on your futurist hat. Where are we going with Echo in the next 5 to 10 years? Yeah,
well, I mean, it's amazing because truly Echo is branching out into so many new directions. So many. It's really hard to kind of put your finger on what is the one or two things, because there's so many things that are exciting that are happening. Of course, people talk a lot about AI, and of course that's going to be very impactful. I think that the AI is where it's really going to help us the most is by taking all of the complex information that Echo gives us that we don't necessarily read or put into a report. So one of the joys that I had was I served on the study. I was the chair of a study section for NIH on imaging. And that, right when I served on that committee, right when AI was really kind of exploding, and there's this thing called radiomics, which means trying to draw as much information you can out of either measurements or features to essentially add to the electronic medical record. The other place that I really hope we go, and there are several of us that are kind of leading this charge, which is we're not going to be diagnosticians with Echo. We're going to be therapeutic deliverers with Echo, I think. There's so much that's going on with sonothrombolysis, which with trying to dissolve blood clots in either the microcirculation or the macrocirculation. There's the gene and drug therapy. There's the augmentation of perfusion. So that my hope is that our technology, our field of Echo is the only imaging modality where we really change. Oh, sure, MRI can align your protons and make them spin, but we really change the physiology of the vasculature and of other tissues of the body with our ultrasound energy, and I think that's where we're going.
Is that really reachable? In other words, I'm thinking of the stumbling blocks of, damage, therapeutic damages to tissues and things like that. I mean, I think it's fabulous, but it is reachable, is what you're saying.
It is absolutely reachable. I mean, if you actually take HIFU, high intensity focused ultrasound, as a learning lesson, it's amazing how quickly HIFU went from this investigational technique that had a bunch of engineers kind of burning and searing steaks in a focal fashion, to essentially the use of that technology for ablating tumors or for ablating portions of the brain that are responsible for motion disorders. There is nothing that makes me believe that we can't do this. And part of it also, Randy, is the fact that there are clinical needs, there are clinical gaps right now. And a really good one, I serve on the Data Safety Monitoring Board for NHLBI for all cell and gene therapy trials. One of the biggest hurdles right now in gene therapy is they have some vectors that work, but they're just not safe enough right now, and everybody is looking for whatever that golden bullet is going to be that's going to increase the efficacy of gene therapy. And I believe that ultrasound is one of those answers.
By turning it on or unleashing it, how's that going to work?
Yeah, so that's part of the beauty, which is you have to understand the process and the biology before you can set it loose, and that's where we're at right now, which is really understanding. But it appears that essentially ultrasound, and especially ultrasound cavitation of these contrast agents, can change the behavior of cells to make them transport some of the gene vectors. So it's not poking holes necessarily into tissues and just hoping things leak out. It's actually an active process where if you vibrate bubbles and you essentially change the surrounding cells to make transport of things go from the inside, the vascular space, to the outside, to where the cells are that need to be modified.
That's phenomenal. That's phenomenal. One of the things that struck me initially and now does all these years is the incredible power of Echo. You can put the transducer down on somebody's chest and in a nanosecond you see what's going on. Early diagnosis and management of patients is dramatic. Does something stand out where you did an echo or were active in an echo that was just like, holy mackerel, this is phenomenal?
Yeah, it's funny that you say that, because I can't come up with an example. I can come up with about 200 of them, and and they're all in the ER. We long time ago we did a study, looking at the use of left ventricular opacification for finding wall motions for for stratifying patients who came in with chest pain. It is absolutely amazing the diversity of pathology that you find when people come in with, questionable symptoms. Is this acute coronary syndrome? And just the number of, Takotsubo cardiomyopathy, dissections by contrast, eosinophilic cardiomyopathy, different forms of non-compaction, just all the different pathologies. So there's no one thing. There's 200, 300 of them that essentially you come in with the echo probe and you look for something and you get completely steered to a new diagnosis.
No, I mean, it is dramatic. It's basically instantaneous recognition of processes that then go to therapeutics that make a difference.
Yes.
Very, very quickly. Jonathan, no other noteworthy memories to share. So it could be events, people, experiences. You've had many. Give me some examples. Events, people, or experiences.
Yeah. Here's why I consider ASE to be like a family. So my very second ASE meeting that I attended, I was a young investigator finalist. And I was nervous as heck. Alan Perlman was the president at that time, and I remember Alan coming in and kind of soothing my nerves before the presentation. But one of the things that I did the night before to just try to relax, because I was incredibly nervous about getting up in front of a large audience and presenting science, is I went out hitting some golf balls with Kevin Way, a buddy of mine, and I got bit on the foot by a spider. I rented some shoes that they had there, back when I guess you could rent golf shoes to go hit balls. I got bit by a spider, and the next day, during the Young Investigator competition, my foot had swelled up to about 3 times its normal size, and I couldn't put a shoe on. And to this day, I'll remember Mary Alice Dilday, who was she was like a mother to the young investigators in those days. She essentially helped me up to the stage, talked me off of the ledge that it was okay to go up onto the stage with a foot this big, not wearing a shoe. And so to this day, I think I am the only young investigator finalist who ever gave a presentation in a very large lecture hall without shoes.
That's amazing. So you gave a quote at the beginning, how lucky I am, and it was from one of your favorite philosophers. You know who that is?
Yeah, that would be Winnie the Pooh.
Right. And it really was that and some other quotes you had. I'll come back to that in just a second. You said one of your quotes was, if I can remember my notes on this, because I thought it was pretty impressive, is that you should never settle for that which is easy and comfortable. Has that been sort of your motto?
Absolutely. I mean, that's the spice of life, right? Why coast when you can be daring? Everybody should be Errol Flynn. Now, maybe I'm dating myself by saying everybody should be Errol Flynn, but really, be swashbuckling, be daring, and it's amazing how rewarding it can be, because it keeps your mind sharp. You go off and learn all of these new things. It's something that I tell the fellows that I've mentored, which is life is just so much more interesting if you're doing things that are not easy, right, and that you have to struggle for. And then you look back and you're rewarded with all those memories. And
you said also that your basically greatest rewards come from being bold and forging new but challenging paths, and you certainly have done that. So you've had an incredible career. And we're looking forward for it advancing. In leaving, what has the American Society of Echo meant to you?
It essentially, in a word, everything. It's been my professional home. It's been my scientific home. It has been a place where people have guided me. People have pushed me. So you asked me, about being bold and being daring. You know what allows you to be bold and daring? People who essentially encourage you. People who say, yeah, that's a great idea, or who want to come hear about the science, right? Not just the people who are pushing you, but the people who are listening and learning and giving you all that positive feedback that the direction that you're going is positive. So that's part of it. The other thing that it's meant is it's really my family. Yeah, this is my professional family. I can't tell you the relationships that I've forged, the career advice that I've gotten, solid career advice, but also just the friendship and the camaraderie and getting phone calls at 10 o'clock at night from Alan Perlman, or being able to, text back and forth with Jim Thomas about the strangest of things, including the meals that he's had that evening, sharing recipes. It really is. It's a family.
Well, you have been a tremendous asset to the society and to the field, and thank you very much.
Thank you, Randy.